This first part will be the discussion on cellular respiration. I'll discuss the knowledge you require to know prior to class. We'll look at a simple overview of the cellular respiration system. Then I'll go back to a detailed explanation of cellular respiration, and then we'll look at alternative pathways. To prepare for class, you need to be able to list the three main stages of cellular respiration, describe the difference between aerobic and anaerobic respiration, including where each occurs, describe the importance of the electron carriers NAD and FAD as found in cellular respiration, describe how NAD and FAD works. In regard to glycolysis, describe where it occurs, what enters into glycolysis, and what are the products of glycolysis. In regards to the Krebs cycle, you need to define and describe where it occurs, what enters into the Krebs cycle, and what are the products of the Krebs cycle. And in regard to electron transport chain, again, where it occurs, what enters into the electron transport chain, what are the products of the transport chain. You need to describe the importance and the role of oxygen as a final electron receptor in the electron transport chain and how that works. Describe the importance of the fermentation pathway in cellular respiration. Describe how that fermentation pathway produces lactic acid. Okay, let's get a simple overview of cellular respiration. In cellular respiration, we take sugar, in this case which is glucose, plus oxygen. The product is water, carbon dioxide, and 32 molecules or 34 molecules of adenosine triphosphate, ATP, which is of course an energy source. That occurs in three steps. It occurs in glycolysis, the Krebs cycle, and the electron transport chain. The process begins by getting this glucose molecule, which enters into glycolysis. As you can see, it is a 6-carbon molecule that breaks down into pyruvic acid, which is a 3-carbon molecule, which means you get 2 of them. This changes into another chemical called acetyl-CoA, acetyl coenzyme A, which is a 2-carbon molecule. The 3rd carbon, by the way, is given off as CO2 gas. The Krebs cycle takes acetyl-CoA and goes through a series of steps and gives us several byproducts. One of the major ones is hydrogen, electrons, and protons. Same as glycolysis, it also gives off hydrogen, electrons, and protons. These hydrogen, protons, electrons, these atoms are harvested and brought to the electron transport chain where they're released. NAD is the electron carrier which does that. We'll explain how that works in a second. So whether it be from glycolysis or the Krebs cycle, the byproduct of these hydrogen ions or hydrogen protons, electrons, is where most energies are actually harvested at the electron transport chain. So we have the hydrogens released, we add to this oxygen, which is the gas we breathe. The result is your ATP. So let's get into each of these individually, and then I'll give you a summarization at the end. Role of NAD. We talk about NAD here, picking up the hydrogens. NAD should be negative. NAD is an electron carrier. You'll also find in the Krebs cycle another electron carrier called FAD. They work the same way. You have your NAD. It's a negatively charged molecule. It will receive an electron and its hydrogen atom. It'll also receive another electron and grab hold of that, and it'll carry with it its hydrogen atom. So what happens, you begin with NAD, you add a hydrogen atom and electron, and it carries with it the positive proton. This molecule can now travel to any place else in the cell and release the hydrogen atoms, which means the proton and the electron. So NAD has a very important function as an electron carrier. It shuffles the electrons and protons from glycolysis and the Krebs cycle to the electron transport chain. If NAD is not working, it cannot transport these hydrogen protons, electrons, and cellular respiration stops. At least at the electron transport chain. Okay, let's look a little more detail at glycolysis. Glycolysis, we said, begins with a 6-carbon glucose molecule, 1, 2, 3, 4, 5, 6 carbons. In the process of glycolysis, energy has to be put in. We have to add 2 molecules of ATP to get this whole process started. And through a series of phosphorylations, moving phosphorus groups, we're going to produce 4 molecules of ATP. Now, so you had to put in 2, we really have a net gain of 2 molecules here. You also have a release of hydrogens. These hydrogen atoms, both electrons and protons, are going to be picked up for the NAD molecule we talked about earlier. They'll pick it up and carry it on down to the electron transport system. But from beginning with a 6-carbon molecule, end up with 2 3-carbon molecules. Each 3-carbon molecule is a pyruvic acid. Now, pyruvic acid cannot enter into the Krebs cycle. It has to be changed to what's called the transitional reaction, where here's your pyruvic acid. You've got 3 carbons here. You've got 2 pyruvic acids, because this is 6 carbon, each of these 3 carbons. You release this carbon and this oxygen, and make CO2. You also receive this hydrogen through NAD again, and it leaves you with what's called an acetyl group. The acetyl group joins with coenzyme A to form acetyl coenzyme A. This is the substance which actually enters into the Krebs cycle. Next up will be the Krebs cycle. It shows pyruvic acid here, changed to acetyl-CoA here. This is a 2-carbon molecule. This simply travels in a cycle. That's what's called the Krebs cycle. And by releasing carbons and releasing hydrogens, we change it from a 6-carbon molecule to a 4-carbon molecule. The process, we produce hydrogen atoms again, electrons and protons. We produce some ATP. Again, more hydrogen to collect more hydrogen here. We also give off carbon dioxide. When we give off carbon dioxide, so actually we exhale carbon dioxide. This is one of those locations from which we exhale the CO2. Okay, I'll stop now. The next part will continue.